Proteins have a wide variety of different functions in the human body, and much of their function can be attributed to their specific structures. The simplest structure of protein is called the primary structure, and this is merely a chain of amino acids, and most times it strictly refers to the number and sequence of amino acids in a more complex protein. The second simplest protein structure is called the secondary structure. And these proteins are either alpha helices or beta depleted sheets. And these shapes are due to the hydrogen bonds between the amine groups and carboxyl groups of each amino acid. These proteins are fibrous in shape and they aid in structure in the human body. The third protein structure is the tertiary structure, which this is the one of the more complex structures. It is globular in nature, and the shape is due to hydrogen bonds and disulfide bonds, ionic bonds, and different interactions between the R variable of specific amino acids. The most complex protein structure is called the quaternary structure, and this can have a whole bunch of different structures put together along with other molecules. A great example of a quaternary protein is hemoglobin. Hemoglobin aids in the transfer of nutrients in the blood as well as blood clotting. Hemoglobin is made up of multiple tertiary structures as well as the molecule heme, which helps with its function. The largest question facing scientists right now is called the protein folding problem. This question is essentially how are proteins able to go from such a simple chain of amino acids to a much more complex structure in a manner of nanoseconds, and is there another structure in the cell which sort of delegates this change of shape? This question has been around since the 1960s, and it still perplexes scientists today.